Good afternoon. I would like first to thank the organizers for inviting me to ISEMPOCON 2022. It's really a pleasure to be here with you today. I'm only regret not to be able to be present physically. And the topic which was given to me is advancement in precision management of soft tissue sarcoma. And it's quite a broad topic at a time where precision medicine is involving a growing number of histological subtypes of sarcoma, as we are going to see. These are my disclosures. And as we all know, sarcoma represents a very heterogeneous group of disease with over 150 different histological subtypes in the most recent WHO classification. Most of these subtypes are extremely rare, and we were able to collect all the cases of sarcoma over a 10-year period in the French NetSarc database with the support of the French National Cancer Institute. The majority of soft tissue and bone sarcoma have an incidence which is inferior to 1 per 1 million per year, over 120 histological subtypes. So it's a wide variety of different histological subtypes. And as you can expect, the majority of these histological subtypes had actually no clinical trial dedicated to the understanding of what could be the best treatment. So the majority of histological subtypes shown here in this list that you cannot read have only phase 2, randomized phase 2 or phase 3 study in light blue, dark blue, and green, only in a minority of cases. So a lot of work ahead of us. A lot of work with the understanding that a majority of tumors have actually molecular alteration, which are hallmarks of these characteristics and may guide the treatment that we are going to give to the tumor, as it has been shown just before by our pathologist colleague. And that's something on which we have based the selection of the treatment in the recent years with some successes. And probably the first success was, of course, gastrointestinal stromal tumor, with understanding more than 20 years ago that these tumors were affected for the majority of them by mutation, which were mutually exclusive on KIT, PDGF receptor alpha, and if absent, other type of mutation which are rarer. So developing in clinical trials KIT inhibitors, which are often PDGF receptor alpha inhibitors as well, led to significant improvement of the management of these tumors, which were extremely resistant to cytotoxic treatment. We know that some molecular subtypes of KIT mutated and PDGF receptor mutated GIST have different outcome with imatinib, but we know even more that actually the majority of patients with GIST have actually a secondary and develop secondary mutation in response to the therapeutic pressure which is given to them. So new drugs are coming to treat these resistant mutation. The phase 3 study Invictus was conducted in fourth line plus advanced gastrointestinal stromal tumor with a comparison between placebo and ripretinib given at a dose of 100 milligram per day. And the randomized phase 3 study was reported in Lancet Oncology, and you can see here the difference in terms of progression-free survival. And even though the statistical plan did not allow to formally compare overall survival, we can see that the overall survival in this subgroup is, in the group treated with ripretinib, is numerically superior. Interestingly, there was an opportunity to dose escalate ripretinib in patients progressing after a standard dose of 100 milligram per day. And this was recently reported at the ESMO Congress, showing that indeed with this strategy, it's possible to obtain some level of tumor control in patients treated with the double dose of ripretinib, which offers the opportunity to have a subsequent line with a double dose of ripretinib in the patient population progressing under 100 milligram per day of ripretinib. In this case, the doubling of the dose was 150 milligram twice a day, and provided, as you can see on this swimmer plot, sometimes prolonged progression-free survival in a substantial proportion of patients. So a drug which is active And this is one of many drugs which were recently demonstrated to be of interest for the treatment of gastrointestinal stromal tumor. In particular, in this difficult to treat subpopulation of with a point mutation of on codon 842 of PDGF receptor alpha GIST, the 842V GIST are notably resistant to the classical treatments which are given in these patients in advanced phase, whether it is imatinib, sunitinib, and avapritinib, a TKI which is able to block selectively this mutation, was developed in phase 1 and subsequently, as we are going to see, phase 2 and phase 3 clinical studies. And the results obtained with avapritinib in the 842V mutant GIST are outstanding as compared to what was reported with imatinib, with a response rate which was inexistent virtually, and median progression-free survival which was close to 2 months. Here you can see that the majority of patients, over 80%, regardless of the dose, are actually responding to the treatment with a prolonged progression-free survival, as you can see here. With a shrinkage of the tumor, which is of a major magnitude, with some complete responses. And that's something which was not achievable in a population of patients which is extremely narrow, because we are speaking here of probably 8% of GIST overall, but the minority of these tumors are actually relapsing. Nevertheless, we now have a treatment which is active for this patient population. The same avapritinib was tested versus regorafenib in locally advanced unresectable or metastatic gastrointestinal stromal tumor. Again, in a phase 3 study, randomized open label phase 3 study, which was reported recently in the GCO, the Voyager study. And sadly, avapritinib failed to demonstrate superiority over regorafenib in the randomized study. But we can see on the survival curve and also on the tumor control rate that the activity of the agent avapritinib in this patient population is not negligible. And actually, even though the trial was negative, this is a study which confirmed that avapritinib has activity in this situation of third-line treatment, simply not superior to regorafenib. Other agents for gastrointestinal stromal tumor, cabozantinib was reported by an ERTC study, Patrick Shosky and collaborators in the CABOGIST study in patients in third-line. And that these were the results of the study. You can see the median progression-free survival, which is close to 6 months, and a median overall survival, which is also of interest. Cabozantinib has activity in this setting, and that's certainly an agent which deserves to be investigated further in this patient population, which is now accessible to regorafenib and ripretinib, as we have just seen. But the observation that cabozantinib is active and in all secondary mutation in a subsequent report is certainly very much of interest. Finally, to conclude on GIST, the registry study reported by our colleagues from Spain provided very important information on the difficult to treat and very rare population of patients with so-called wild-type KIT PDGF receptor alpha GIST, where no treatment is recommended. These tumor can have indolent behavior, but sometimes are progressing and are therefore difficult to treat. These investigators tested regorafenib in first-line treatment of these difficult to treat tumors, and you can see here that tumor control was observed in a large proportion of the patient included, with a median progression-free survival which was close to 9 months, as we can see here, and actually a tumor control which is encouraging in a population where no treatment was demonstrated specifically to be active so far. All right, so we jump from the paradigmatic model of the GIST with KIT and or PDGF receptor alpha kinase mutation to other kinase mutations. And of course, the classical vemurafenib basket study, which was reported some 5 years ago now in the New England Journal of Medicine, offered the possibility to look at what was active in the population of connective tissue tumors with BRAF mutations. And actually, there were very few connective tissue tumors, but the study was later expanded to look at a broader population of patients with Erdheim-Chester disease and Langerhans cell histiocytosis, showing in this subsequent report a large proportion of patients with tumor control. Actually, the majority of patients achieve tumor control in this setting. No, a classical sarcoma histotype, I would say more frequent sarcoma histotype, were included in this study. Other studies have been reported showing responses in undifferentiated pleomorphic sarcoma affected with BRAF mutation, which is, of course, extremely rare. The same patient population, histiocytic in a close patient population, histiocytic sarcoma with MAP2K1 mutation were shown to be efficiently treated with inhibitors of MEK, showing the importance of this pathway in these very rare sarcoma subtypes. Again, an example where we should be aware of the nature of the mutation, but that sometimes clinical trial can be difficult to do simply because of the rarity of the tumors in these cases. All right, so what about sarcoma with translocation? It started so quite long ago, actually, with a discovery by Bob Mackey and collaborators and several others case reports that the agent imatinib was also active on locally advanced or metastatic dermatofibrosarcoma protuberance, which, as you know, is associated with a fusion gene juxtaposing a collagen gene with a growth factor of the PDGF family. And actually, this was followed by a rapid approval from the health authority. Probably would not obtain it today as quickly, followed by phase 2 study conducted on both sides of the Atlantic, which were merged and were presented in GCO, showing a significant proportion of patients with tumor control in this patient population, which was notably difficult to treat. That's a rare event to have a metastatic relapse in this disease, but to have a treatment which is active sometimes for more than a decade in personal patient experience is something which is extremely important. Not really sarcoma, but locally aggressive connective tissue tumor, PVNS, are also affected with similar mutation of collagen with fusion gene juxtaposing collagen, one of the collagen gene, with a growth factor, CSF1. This is giant cell tumor of the soft part with a typical mutation. And from a single case study to a retrospective to a prospective study, it was clearly demonstrated that tyrosine kinase inhibitors blocking CSF1 receptor were active, providing tumor shrinkage for some patients, patient relief. For this difficult to treat tumor, was a surgical oncologist or the orthopedist tells that once the tumor has relapsed, it's very difficult to propose a treatment which can be useful for the surgical treatment, which can be useful for the patient. Interestingly, with the understanding of the biology, some trials were conducted showing anti-tumor activity of this pathway by targeting this pathway. The emactuzumab phase 1 study demonstrated a high level of tumor control with an anti-CSF1 receptor antibody. The nilotinib study, which was conducted in the country where I work together with Australia, the Netherlands, and Italy, also confirmed some level of activity, even though the shrinkage was much less pronounced. Interestingly, this approach of tyrosine kinase inhibition was conducted with an agent which is specifically active on CSF1 receptor, pexidartinib, which was shown in a randomized phase 3 study to be able to provide a significant level of shrinkage as compared to placebo, which is also long-lasting, and which led to the approval of this agent in the US, sadly not in Europe, where no treatment is registered for this indication yet, even though obviously, as shown by Dr. Silvia Stacchiotti from the Istituto Tumori in Milano, some of these tumors can be extremely mutilating or debilitating, and that tumor shrinkage can be obtained sometimes very spectacularly. That's only another example of what we can achieve by targeting fusion genes which are actionable. Another example also conducted by the ERTC with first Dr. Patrick Chowsky, well, it was recently updated actually, with crizotinib in inflammatory myofibroblastic tumor, super rare entity in a country of 60 million people where I live, about 15 to 20 new patients per year. So it's really something which is quite exceptional, affected with ALK, ROS1 or NTRK mutations, most often ALK, and crizotinib was for this reason, tested in this patient population, providing prolonged progression-free survival and sometimes prolonged overall survival. Also, the personal experience of a patient who had brain mets at the beginning and who is still there trying to get pregnant 7 years after the initiation of the treatment. She's now stopped the treatment, of course, is exceptional and shows the value of this treatment and the activity of this treatment in a fraction of these patients. We cannot escape the topic of NTRK inhibitors, which were demonstrated to be very active in tumors bearing TRK fusion genes in adults or in children, with two agents currently registered, larotrectinib shown here and entrectinib also shown in a mixed bag population of patients, including sarcoma. And with sarcoma, and probably the incidence of NTRK fusion genes in sarcoma in general is close to 1% from a relatively large experience conducted in a molecular laboratory where in the institution where I work. This is rare, but the treatment is very efficient, as you can see here. Very efficient with a duration of response, which is close to, as you can see, 15 months, and the duration of median progression-free survival and overall survival, which is close of what we observed in GIST some 20 years ago. These slides were taken from a presentation by the Dr. Gunder some 4 years ago and are so impressive that I always show it to demonstrate really the benefit of that can be obtained with some patients. And sadly, this is not available everywhere around the world, including in my country where this is not reimbursed for adult patients, but the activity is really very much impressive. Finishing with translocation, there are more difficult targets to target, and typically fusion genes generating transcription factors which are aberrant are difficult to treat. Ewing sarcoma are more frequent. There is now agents which try to target specifically the pathway which are activated in this translocation, the ETS pathway, and TK216 was reported to have 2 CRs in a phase 2 study, which was reported recently in oral, and that's something to be investigated further in the future. All right, so it works in mutation, translocation. What about amplification? Amplification of MDM2 and CDK4 are frequent in a variety of sarcomas, liposarcoma in particular, leading to the cell cycle activation. And in this case, attempts to block MDM2 overexpression were conducted in several clinical trials, phase 2 studies, which all very consistently demonstrated reactivation of p53, but a modest impact on progression-free survival alone. And the same was observed actually when one tried to target CDK4, which is amplified also with palbociclib here, a signal of activity, no doubt about it, but which is not long lasting. So probably the next step is going to be combinations. And actually there is a phase 2 ongoing in this country and accruing quite nicely, which should provide results of interest in the coming year after a phase 1 study, which also shows a signal of activity. Other examples, yes, there are other examples of targeted treatment. One of the great examples is the loss of tumor suppressor gene TSC1 and TSC2, which is frequent in the super rare PEComa type, malignant PEComa and PEComa. The malignant form of PEComa is a difficult to treat disease. In this retrospective study conducted by our colleagues from Istituto Milano, sorry, the best results were obtained actually with an mTOR inhibitor, which makes a lot of sense biologically by targeting the pathway downstream, which is activated by the loss of the tumor suppressor gene. And one drug has been approved and reimbursed in the US so far with a liposomal form. This is a pathway which can be targeted in rare tumors. Other agents, yes, there are other agents in a variety of different tumor types. I think it's important to mention the epigenetic next generation target with epithelioid sarcoma, which is, as you know, affected sometimes with the loss of INI1, leading to the activation of EZH2. And tazemetostat is able to block EZH2. And with this, in first-line treatment of epithelioid sarcoma, a very rare entity, probably with an incidence inferior to 1 per million per year, the majority of patients did achieve tumor control or stabilization. And this is a drug which can be given in this setting and which is currently being explored in a prospective study. It's also important to quote in the mixed bag the activity of sorafenib, very elegantly demonstrated by our colleagues from the US. Dr. Gunder, first author, showing superiority of sorafenib over placebo, but still in desmoid tumor, without a true understanding of the exact biology behind that. Placebo was inducing tumor shrinkage in about 20% of the patient, but this was much better with sorafenib, which enabled to induce tumor control in the majority of tumors. Right, I would like to conclude by saying a few words on immunotherapy of sarcoma, which used to be the first treatment given systemically to patients with advanced sarcoma, with signals of activity in osteosarcoma and with limited activity in single agent or combination treatment in the first phase 2 study or randomized phase 2 studies which were conducted. We should not, however, desperate because we now understand by pulling the tumors together in a very nice retrospective translational study that probably the patients responding are those bearing tertiary lymphoid structure in the primary tumor, the red group here. And this was confirmed subsequently in a recently reported in oral format study exploring pembrolizumab, confirming the activity of pembrolizumab in the 15 to 20% proportion of patients with advanced sarcoma with tertiary lymphoid structure. And then there is another group of sarcoma with where immunotherapy is quite active, which is the rare chordoma and alveolar soft part sarcoma, and several studies have now been demonstrating this very nicely. Probably one of the very exciting observations in the recent year, and I would like to conclude with that, is the observation that modified T cells to express T cell receptors recognizing antigen expressed at the surface of sarcoma cells, synovial sarcoma, or myxoid round cell liposarcoma, were demonstrated to be active in single arm studies reported this year at CTOS, with tumor shrinkage, which is impressive for patients with synovial sarcoma receiving cells recognizing MAGE-A4 in an HLA-A2 context. And the same was reported in the same CTOS using another cell modified, recognizing this time NY-ASO1 in a smaller subgroup of patients with myxoid liposarcoma with responses which were of an important magnitude and durable for some patients. So certainly something which needs to be investigated further, but altogether we have a lot of work to do. That will be my conclusion. With 150 different histological subtypes, we are progressing in the field of development of targeted treatment on molecular alteration, but we do not always understand what is driving the response. Sometimes we have an efficiency which is unpredictable. We have good examples and less good examples. Probably immunotherapy is the next generation of treatment which will be developed in a subset of patients. All this calls for international collaboration to develop clinical trials. Thank you very much for your attention and happy to take questions.
Yeah, that was an excellent talk. I would like to know your viewpoint on the use of pasopanib. We have few studies from India and also from outside that pasopanib is effective in synovial sarcomas and in second line. So how do you think, do we need to have a target to be identified, or we can use it just without a target or without NGS?
I think that pazopanib is indeed demonstrated to be active in synovial sarcoma. You're absolutely right. When we look at the subgroup analysis of the standard clinical randomized clinical trials, the number of patients in the synovial sarcoma group is low. But we know that by experience using close agent, which is regorafenib, the mode of action of this class of agents is very specific for specifically active in synovial sarcoma with hazard ratios as compared to placebo, which are under 0.2. So yes, definitely without any more than the molecular demonstration of synovial sarcomas, is definitely a good agent for this subset.
And one more question due to the paucity of time. For soft tissue sarcomas, is it better to do the NGS on the primary, or when they develop a metastasis, should we do a biopsy then and do an NGS?
On the primary would be my favorite option because it's easier. And second, you are certain to have a driver molecular alteration with all tumor cells. Now, that doesn't exclude, as mentioned by Paolo Deitos a few minutes ago, that of course additional mutation can be found in the METs, but primary would be my preferred option.
And we should be, if in ideal situation, we will be doing it in the first line. Means we will not wait for the metastasis to come. We should do it in the first workup itself.
Exactly.
Okay. Due to paucity of time, over to Dr. Jyoti Bajpai.
So thank you very much. Indeed, it was a fantastic talk, fantastic session, and we would actually love to ask many questions. But as Sujit said, that we have to respect the time, so I will send you all those questions. And thank you for joining us.
Happy for that. Thank you very much.